I am Marta Cosin, a postdoctoral fellow at IS Global, and I am presenting a study on the impact of exposure to secondhand smoke during childhood on DNA methylation. Exposure to secondhand smoke has been associated with a number of health outcomes, including poor lung function and respiratory conditions in children, and there is a need to understand the biological impact of this exposure during childhood and to identify biomarkers that may predict future disease risk. Unlike with maternal smoking during pregnancy, very few studies have investigated postnatal exposure to secondhand smoke and offspring DNA methylation specifically. Therefore, our objective is to conduct epigenome-wide meta-analysis to identify associations of postnatal exposure to secondhand smoke and childhood blood DNA methylation and its interaction with in-utero exposure to smoking. To do this, we use data on prenatal and postnatal smoke exposure and childhood blood DNA methylation from almost 3,000 individuals from 7 cohorts within the PACE consortium. We perform linear regression models and inverse variance weighted fixed effects meta-analysis. Lambdas range between 1.1 and 1.3. The results of the two models explored so far are summarized using Venn diagrams in this poster. In the first model, we found that more CpGs were associated to maternal smoking during pregnancy than to any postnatal exposure to secondhand smoke, as expected. Moreover, only methylation at 4 CpGs was significantly associated with both exposures. In the second model, that includes the interaction component, we found that the effect of postnatal exposure to secondhand smoke on child DNA methylation can be modified by the in-utero exposure for a number of CpGs, in particular 64. There was no overlap with CpGs influenced by the exposures alone, suggesting that this group of CpGs need both exposures to experience a change in methylation levels. Interestingly, many of the CpGs identified were already associated with maternal smoking during pregnancy or own smoking, and are associated with cancer, inflammatory diseases, or aging. Please visit our virtual poster and use the chat application if you would like to discuss our results and the next steps with us.